Class is now starting. All attendees are in listen-only mode.
Good morning, everyone. Welcome to the December Impact webinar for 2019. This is Rebecca Bailey, and I will be your moderator for today's webinar. Today, the webinar will be presented by Dr. Eric Sobey. Dr. Sobey is a professor in the Department of Pharmacological Sciences at the Icahn School of Medicine at Mount Sinai in New York. Dr. Sobey obtained a bachelor's degree in biomedical engineering from Duke University and a PhD also in biomedical engineering from Johns Hopkins University. His research focuses on obtaining new insights into cardiovascular disorders and potential treatments of those disorders through a combination of physiological experiments and mechanistic mathematical modeling. Since 2006, Dr. Sobey has directed an NIH-funded laboratory at Mount Sinai that applies engineering and mathematical modeling approaches to unresolved questions in drug development related to both efficiency and safety of cardiac drugs. His group uses mathematical models of heart cells and tissues to generate novel predictions of drug effects and experimentally test those predictions wherever possible. Dr. Sobey will be taking questions at the end of the webinar. You can write your questions into the questions pane on your GoToWebinar side panel at any time during the webinar, and then we will be addressing all of the questions at the end of the webinar. And with that, I would like to hand over the webinar and welcome Dr. Eric Sobey.
All right. Thank you, Rebecca, for the very kind introduction. And if you have any problems with the audio, then please put something in the chat box so that the organizers can be aware of that. We can try to fix that. But otherwise, I will just go ahead and get started. So today, as the title slide indicates, what I intend to talk about is how in my group we've been combining mechanistic mathematical modeling with machine learning approaches in order to predict cardiotoxicity and streamline drug development. So I'm going to start with an introduction to what I'm talking about with these mechanistic models and what I'm talking about with these machine learning approaches and how we try to, and an overview of how we try to combine them. So the QSP, or quantitative systems pharmacology style models that we use in my lab are primarily of heart cells and heart tissues. And I think the highest level way to think about some of these models is by considering what the inputs are, what the mechanisms are, and what the outputs are. So the inputs that we're talking about in these models are the things that in the laboratory you would do to manipulate a cell or to perturb a cell. You would electrically stimulate it. You might modify the temperature, or different experiments might be done at different temperatures. You might modify the ionic concentrations. And then, of course, because we have pharmacology interests, you might add drugs. And the drugs in this case are mostly influencing these heart cells by blocking ion channels. The mechanisms that these models simulate are the ionic currents that can go through ion channels, through pumps and transporters. And then, of course, we also have to consider ionic homeostasis, whether the concentration of a particular ion is going up or down, and what the processes are that regulate that ionic concentration. And when we combine the inputs plus the mechanisms, then we get the outputs of these mathematical models. And the outputs that we're primarily concerned about are the action potential in the cardiac myocyte, voltage as a function of time, and the calcium transient in the cardiac myocyte, how intracellular calcium changes as a function of time. And here I've indicated some of the numbers that we try to extract, some of the features that we try to extract from these time course measurements: action potential duration and calcium transient amplitude. And the question is, why do we care specifically about these? Well, the electrical activity, the action potential, is what it produces the electrocardiogram in patients. So that correlates at the cellular level directly with something that you can measure in people. And then the intracellular calcium is a function of time. This is the signal that enables contraction in the cardiac myocyte. So this also directly correlates with something that you can measure in people, as well as something that can be perturbed and can be dysfunctional in disease states. So this is how we can think about these mathematical models that we're dealing with at a very high level, just in terms of the inputs, the mechanisms, and the outputs. If we want to dig a little bit deeper into the actual equations underlying these models and what they what they do and how we use them and how we implement them, these are that's what this slide is for. These models simulate the ionic currents, intracellular ionic homeostasis, and these models started with the these are out of the tradition of Hodgkin and Huxley, and then in the cardiac realm, starting with the work of Dennis Noble at Oxford over 50 years ago. So we've been building on a long history of these mathematical models. We represent our cardiac myocyte like this, where you have an extracellular membrane, you have intracellular organelles, most importantly the sarcoplasmic reticulum, and then you have different ion channels, different pumps, different transporters in the cell membrane that influence the electrical and contractile activity of the myocytes. And on average, we're dealing with somewhere in the neighborhood of 10 to 20 different discrete molecular species. And these are proteins that we can identify with genes, somewhere in the neighborhood of 10 to 20 channels, pumps, and transporters. This altogether, this gives us a system of somewhere between 20 and 60 ordinary differential equations that we have to integrate and solve in order to calculate and predict what the electrical activity of these myocytes is going to be. And the drug effects are simulated by either reducing activities or enhancing activities, depending on what's appropriate for a particular drug. And the sorts of outputs that we get from this and the way we need to compute this, this is an example of a particular ionic current, which is computed as a conductance times a couple of variables to describe the opening and closing of the ion channel, the gating of that ion channel, times the driving force. And then the action potential and the intracellular calcium transient that I mentioned on the last slide are calculated. They have differential equations that represent voltage and calcium, which depend on what the processes are that might drive it, that each variable in one direction or the other direction. And of course, if we have a system of 20 to 60 ordinary differential equations, then from a mathematical point of view, what that means is we have 20 to 60 outputs, right? There's going to be 20 or 60 the results of the numerical integration. But as I mentioned on the previous slide, we care mostly about the action potential and the intracellular calcium. And the reasons for that are that, first of all, that's something that you can measure and experiment. And I'm not going to talk so much about our lab's experimental work today, but that's a really important part of what we do is we do the experiments to test our predictions. And then, as I mentioned on the previous slide, part of the reason we care so much about the action potential and intracellular calcium is that you can directly correlate those cellular signals with what you might see clinically in people. So that's the quantitative systems pharmacology part of what I'm going to talk about, just to give a general introduction of the types of models that we're dealing with. What about the machine learning part? That's I think what the exciting part is about what we're doing is how we're trying to combine these different approaches. Machine learning has been used in drug development. It's becoming more and more popular. But based on the reviews I've read and people I've spoken to, I would say there's really been two primary areas in which it's made an impact, its greatest impact so far. One is an analysis of drug chemical properties. This is just an example slide from a recent review article in Drug Discovery Today, and how you can use the machine learning algorithms and you can encode the different chemical properties of your drugs and learn associations between different drugs and maybe generate predictions about what particular chemical properties might give you a good outcome or a bad outcome. And then a second major area in which machine learning has been used a lot in pharmacology has been to analyze patient data and to perhaps group patients on the basis of the clinical data that you measure to say, which patients are going to be the healthier ones and which patients are going to be the less healthier ones, even if they all have the same nominal diagnosis. And this is a very influential paper from a couple of years ago in Circulation, looking at a particular type of heart failure. And from a machine learning analysis of the clinical data, was able to categorize the patients into 3 different groups that had different outcomes. So these are both examples of machine learning that's being used in drug development, but just from the standpoint of taking a lot of data that are out there and crunching those numbers and statistical analysis of those numbers. The question we want to address is there any benefit in combining the machine learning approaches with the mechanistic modeling that I already described in the last two slides. And that's what I hope to convince those of you who are here that we do think there's a benefit in doing this, and we think that we have a way of combining the two that is going to hopefully lead to insights. We have some insights, but a lot of it, of course, is a work in progress, and something that we're working really hard to try to continue to improve. So as I was preparing for this webinar, I did a little trip down memory lane. In some ways, this is not a new idea at all to say we want to combine statistical approaches with mechanistic approaches that consist of systems of ordinary differential equations. It's not a new idea, that I'm putting forward here in 2018, and it's not even a new idea that I've had. This is a slide that I put together back in December 2009, so 9 years ago. And this was one of the ways that I was conceptualizing at the time systems biology. I'm not going to go through all the details here because I want to get to the results, and this is a very busy slide. But just in a very broad sense, what I was trying to argue with this slide was that over on the left-hand side, you have omics type analyses that are a big part of systems biology. And you can see how dated this is because I had microarrays as an example of an enabling technology of systems biology. And of course, no one uses microarrays anymore. And then I said that the omics tradition is one part of systems biology, and the physiology tradition is another part of systems biology. So on the right-hand side, you have the part that's small scale and mechanistic. On the left-hand side, you have the aspect that is large scale and high throughput, but less mechanistic. And if you can combine the two, then you were really doing cutting-edge systems biology. As I mentioned, a lot of other people were expressing similar thoughts around that time. So this was not my idea by any stretch of the imagination. But this is just to indicate that we and others have been thinking along these lines for quite a while about bringing together the power of statistical analyses, the power of bioinformatics, combining that with the power of mechanistic-based investigations. So this is my concept, nearly a decade ago, and we've been working on that since then. And now we have a sort of conceptual way of thinking about how we combine these that looks something like this. That I think is maybe a little bit more precise than what I showed on the last slide, and hopefully is going to lead to some advances and really help us better understand how to develop drugs. We start with quantitative systems pharmacology of the types of heart cells that I mentioned. Then when we run the simulations, we build populations of action potential. So we simulate heterogeneity in the population in order to generate a variable response. And in this particular example here, I'm going to get into the details later, but we add a perturbation, and in some of these cells it might cause an arrhythmia, and some cells it might not. From these population results that we simulate over here on the left, we plug those population results that incorporate variability into the machine learning algorithms to try to differentiate between classes in terms of the responses that we get. So this is one aspect of the combining of the quantitative systems pharmacology, mechanistic-based simulations with the machine learning approaches. But then the really important part that we have now, which we maybe hadn't thought about so much back in 2009 on the previous slide, is feedback from one to the other. And the reason the feedback is important is this can allow us to address really important questions that, by doing this iteratively and by closing the loop, so to speak, we can address questions such as, well, what mechanistic differences explain good classification or poor classification? So if you look at your population here and you try to classify them into group 1 and group 2, sometimes you're going to get it right, sometimes you're going to get it wrong, and that can give you clues for what you can look at in the mathematical models to say what can differentiate the cells that are easy to predict from the cells that are more difficult to predict. And then the other question that I'm gonna mention that we're really focusing on a lot at the moment is which simulation protocols can improve prediction. So you can study a heterogeneous population under one condition, and looking at it under one condition might give you poor classification with your machine learning. But if you look at the same population under a different condition, it might improve your classification, and that's really important because that can help you prioritize your experiments, and that can also give you important insight into, well, what was it that's bad about one experimental protocol and better about another experimental protocol, And so it gives you allows for prioritization, as well as giving you insight into what might make some experimental protocols better than others. So with that introduction on how we conceptually look at the combination of the quantitative systems pharmacology and the machine learning, we can get into some more of the details and get into some of the actual studies that we've been performing recently. So once we have this concept of combining the two, we can address particular questions. One is, can we streamline and improve testing of drugs for potential proarrhythmic effects? Another question we can address is, can we correct for limitations of experimental models? And then the third important question is, can we distinguish between susceptible and resistant patient groups? And these three questions, as I posed them, map very nicely onto three studies that are either completed or underway, and that can allow me to actually present an outline of what I'm going to talk about in terms of the detailed analysis. One study, which we published a couple of years ago in CPT, is computational prediction of proarrhythmia. That's the first thing I'm going to talk about. This was work done by a MD PhD student in my laboratory, Megan Cummins Lancaster, who has subsequently graduated and moved on to Vanderbilt to study cardiology. The second study is about how we can predict drug effects across cell types. This is work done by Jingxi Gong, who's currently a PhD student in the laboratory, and it was published earlier this year in NPJ Systems Biology and Applications. And then the third story I'm going to talk about is work that is in progress, done by another PhD student in the laboratory, Mira Varshnaya, along with Shuiyan Mei, whose picture I don't have, and this is unpublished work in progress. So in the remainder of the webinar, I'm going to go through these three stories relatively quickly, so I can cover everything in time to have questions. But it's going to be these three questions we can address with this combination of machine learning and QSP. So I'll start with computational prediction of proarrhythmia. Again, the work of Megan, published in 2016. The question that we were trying to address in Megan's study is drug-induced torsade de poids. And this is those of you who work in safety pharmacology are very well familiar with this, I'm sure. This is a very serious adverse event that can be caused by blockade of cardiac ion channels, especially a channel that we call hERG. That was the old-fashioned gene name for it. It encodes an ionic current you can measure called IKR. And blockade of hERG can prolong action potentials and cause cardiac arrhythmias. So the general model that we can think about with respect to how drug-induced torsade occurs looks something like this. You start with hERG block over here on the left. The hERG channel happens to be very promiscuous. It's got a wide mouth. It can bind a lot of different drugs. If you get hERG block, you're going to prolong your action potential at the cellular level. So the action potential measured with the drug is longer than the control action potential. That's going to lead to prolongation of the QT interval in the electrocardiogram, and that's going to very dramatically increase your risk of developing this polymorphic ventricular tachycardia known as torsade. And this model works pretty well most of the time. You can, if you know if a drug blocks hERG, you have a pretty good sense of whether that drug might be torsadogenic or not. But there's some complicating factors, and there are some examples of when it doesn't work it's not quite so simple. Drugs can block multiple ion channels besides hERG. Arrhythmias can arise without getting substantial action potential prolongation. And effects that occur both at the cellular level and at the tissue level can contribute. So hERG block doesn't always lead to action potential lengthening, and action potential lengthening doesn't always lead to QT prolongation or doesn't always lead to torsade. So as I mentioned, this model for hERG block leading to torsade works pretty well. It works a lot of the time, but there's a lot of examples where it doesn't work, and that's what we wanted to address using this combination of machine learning and quantitative systems pharmacology. So the overall outline of our study was to combine the bioinformatics side of things with the physiology side of things in order to develop a better classifier for whether drugs might cause torsade or might not. We started with data from the literature on how a whole series of drugs blocked different ion channels that are important for heart cell function. And what we had to work with was a table of IC50 values. I'll show some examples in the next slide coming up. We plugged those numbers into our mechanistic cellular quantitative systems pharmacology models to predict what the physiological outcomes would be when these drugs were applied to cells. We also got some information from a toxicity database to say which drugs might be torsatogenic and which drugs might not be torsatogenic. Then we took this toxicity database, clinical data, combined that with the results of our simulations using machine learning algorithms. And through this combination, we were able to obtain improved identification of potentially cardiotoxic drugs. So this is the overview of the strategy that we used in this study. As I mentioned, we developed our algorithm based on multi-channel block. This is showing you an example of what the input data was, where we had a whole series of drugs. And what we had in terms of how the drugs worked at the molecular level are IC50 values for 3 important cardiac ion channels. This hERG channel, for those of you if you're not familiar with this, the details don't really matter so much. But for those of you who might work in this area or something similar, what we had was data for the hERG channel, the rapid delayed rectifier potassium current, NAV 1.5. This, of course, is the primary cardiac sodium current. CAV 1.2 is the cardiac L-type calcium channel. We had IC50 values for how the different drugs affected these 3 channels, and we knew whether the drug was considered a positive for risk of torsade or was considered a negative for risk of torsade. Overall, we had 67 unique drugs blocking potency for the 3 ion channels and clinical torsade risk. And what we did in these simulations is we generated a large set of pseudo data. We decided to take an agnostic approach where we said, for each drug, what do we want to quantify from these time courses? And we said, well, let's quantify anything that we could take, that we could do in the laboratory. If we had the cell in a dish and we added a drug, what are all the metrics, what are all the features that we would extract from those time courses? So this is indicating over here the things that we measured based on what classically you would do as an electrophysiologist or a cellular physiologist when you had these time courses. We also took multiple models, paced the cells at a lot of different rates under several different experimental conditions. Again, inspired by what you would do if you had the cell sitting in a dish and you wanted to test it. So this gave us this large set of pseudo data where we had 67 unique drugs. We had 86 drugs for the standpoint of a classifier, because some of them we had multiple sets of ion channel block data. We simulated both measurements in that case, and then we had many measurements that we were extracting after we simulated the effects of each drug. So overall, we had the 331 metrics from the 67 unique drugs that we were simulating. So what do you do when you have a large set of pseudo data, like simulated data like this? Well, one thing you can do is you can just show the data in a heat map. This heat map looks nice, but it doesn't really tell you that much. It's really hard to get information out of this. And when you have a large set of pseudo data like this is where the machine learning algorithms can be very helpful. And this is what bioinformatics or machine learning type approaches, statistical modeling is good for. So that's what we did. I'm skipping over some of the details here because this is published, but we simplified the data with principal components analysis, and then we used support vector machine in order to develop a classifier. And what we got in the end was an improved classifier compared with whatever the standard is, what the standards are in analysis for cardiotoxicity right now. So what we're showing here is the receiver operator characteristic curve, also known as the ROC curve, where the true positive rates on the y-axis, false positive rates on the x-axis. And if you have a classifier that is completely useless, where you're just guessing for each drug, is this drug going to be a torsatogenic drug, or is it going to be a safe drug? Completely useless and random classifier would fall somewhere along the line of identity. So what you want to see with this ROC curve is how high does it get above this line of identity? And the higher it gets, the better it is as a classifier. And what you can see with this first curve, this is what you get if you just look at block of hERG, is that you get good classification. So this is what I was saying before, that looking at whether a drug blocks hERG tells you a lot about whether that drug is going to be torsadogenic, but it doesn't tell you everything. If you just look at how a drug influences the action potential duration, and this would be equivalent clinically to looking at QT interval, so this is the information you get out of a thorough QT study, for instance. If you look at that information, you get a slightly better classifier than you get if you only look at hERG block. So this is also very useful. But if you look at our classifier, where we put everything together based on the simulations that were done under multiple conditions and all the different features that we're extracting from these simulated experimental time courses, then you get a classifier that's even better. So the way that you quantify this is by computing the area under this curve, and we were able to go from 0.81 all the way up to 0.96. So a considerably better classifier for looking at whether drugs cause torsade or don't cause torsade. So this was giving us superior arrhythmia risk for real drugs. But the really important thing to emphasize here is that it isn't just about getting the prediction. What do you get from doing the mechanistic modeling, the QSP modeling? And the argument is you get something that's a lot better than just prediction, whether a drug is good or bad. These are things that that are in the study that I'm skipping over so I can get to some of the newer unpublished work. But it's an important point to make that we were able to simulate under many different conditions. And one of the things we saw when we looked at dose dependence was sometimes we only got arrhythmia risk at high plasma concentrations. There are a lot of drugs that look safe when they're at a normal therapeutic concentration. At a higher concentration, that's when you can see that they might be dangerous. And of course, they're probably only dangerous for the people that have personal PK such that they're probably going to experience higher concentrations in their hearts than other people will. We're also able to do some precision predictions. We simulated some heterogeneous population in order to look at which individuals were greatest risk for drug-induced torsade. And the unpublished work I'm going to show at the end is an extension of that work. And then the other really important thing is experimental prioritization. The simulations were able to tell us which ion channels should be assessed, which assays should be conducted during the in vitro physiology experiments. So I'll show an example of that last one, how we map the classification to physiology and how that tells us a way to experimentally prioritize. We did our initial classifier based on all of the results that we got from our simulations, that very large set of pseudo data, 67 drugs by 331 metrics. The question is, can we just look at individual physiological metrics or combinations of physiological metrics and still get good classification? So Megan did an analysis where she systematically determined the most informative pairs of uncorrelated metrics. And the one that came up at the top of the list was intracellular calcium and action potential duration in one particular mathematical model simulated at a particular pacing rate. And this is one of my favorite slides from this study. This is really cool. What we're showing on this particular plot is diastolic calcium on the y-axis, action potential duration on the x-axis, and all the bad drugs, all the dangerous ones are shown in red, and all the safe drugs, the blue ones, are shown in blue. And what you see here is that for these drugs over here, which cause very dramatic action potential prolongation, so as you get to the right side of this along the x-axis, as you move far to the right, the drugs that cause dramatic action potential prolongation are uniformly dangerous. That means that looking at QT prolongation is helpful, right? So we're not trying to argue against that conventional wisdom about hERG block or against that conventional wisdom about doing a thorough QT study, right? The ones that cause dramatic prolongation are the dangerous drugs. But what we see is in this middle range, the ones that cause just a little bit of action potential prolongation, and these are zoomed in over here. So that's the inside is a zoom in of the box here. If they're in this range where they only cause a little bit of action potential prolongation, they might be dangerous or they might be safe. And so we picked a couple examples here. Dofetilide and piperacillin just happen to produce exactly the same amount of action potential prolongation. And dofetilide is in the dangerous group and piperacillin is in the safe group. So if you were just looking at the action potential, you wouldn't be able to tell which one was which. However, if you also know the intracellular calcium, then you can distinguish all the ones that cause an increase in calcium, not all, but for the most part, they tend to be more dangerous, and the ones that cause a decrease in intracellular calcium tend to be safe. And so you can draw a boundary between the dangerous category and the safe category if you have both the action potential duration and the intracellular calcium, whereas if you just had the action potential, you wouldn't be able to tell the difference between the dofetilides and the piperacillin. So we thought this was a really cool result and an important result because it tells you what the value is of looking at the mechanistic model in terms of how it can help you with experimental prioritization. So measuring action potentials and calcium is better than only measuring action potentials. And that's the key takeaway of this study, is that the simulations do not only assist in classifying drugs, they can also indicate which assays are the most helpful and which ion channels contribute to toxicity. That was another aspect of the study that I didn't get a chance to talk about, but we had more figures in there that talked about which ion channels you should be measuring the block of. So the way to summarize this study, right, before I move on, is to think about how models are how mathematical modeling is being used right now for this question of prediction of toxicity. We're not the only ones thinking about this. There are lots of people thinking about this. In fact, there's an initiative going on called the Comprehensive In Vitro Proarrhythmia Assay that is a partnership between the FDA, academic laboratories, and pharmaceutical companies. And the CIPA, or the Comprehensive In Vitro Proarrhythmia Assay, can be conceptualized like this, where you take measurements of ion channel block on multiple ionic currents, you plug those numbers into the simulations to see how the different drugs are going to affect the action potential, and then you have a comparison between what you get out of the simulations with what you get in terms of cellular physiology. And the cellular physiology in this case is going to be effects of the drugs on human stem cell-derived ventricular myocytes. So this is how CIPA is currently being thought of and how it's currently being used. But I would argue that we can maybe make the modeling, maybe give the modeling in CIPA a more prominent role. And what I mean by that is that you start with the data on the ion channel block. You use those data on the ion channel block to predict what's going to happen at the cellular level, but you don't then just compare it to what you get experimentally. You want to have feedback from what happens in the simulations can inform which ion channels you should be measuring. There might be some that you didn't think about at the beginning that turn out to be really important, and the simulations can tell you that. And then the simulations can also tell you what you should be doing in terms of the experiments. So that specifically what I just showed a couple slides ago is that if you look at action potentials plus calcium, that's going to give you better classification than if you only look at action potentials. And that came about because we had this combination of the machine learning with the mechanistic modeling simulation. So I would argue that the modeling, if you use it in the right way, it can have a more central role. It can inform your experiments in a way that can be really helpful. So this is how CIPA is currently envisioned. I talked about how the ion channel block measurements can go into the simulations, and then I just briefly alluded to the fact that the simulation results are going to be compared with the experiments that are going to be performed in the stem cell-derived cardiac myocytes. So now I want to focus on this part of it. What about this comparison between the simulation results and the stem cell-derived cardiac myocytes? So now going back to the outline, I'm transitioning from study 1 to study 2. This was the paper that we published at the beginning of this year by Jingxi Gong, who was the lead author on this study. How do we predict drug effects across cell types? This slide shows what drug effects might look like in a human adult ventricular myocyte and what they might look like in a stem cell derived myocyte. And as I mentioned, comparing the effects in one cell type with the effects in the other cell type is one of the three pillars of the CIPA initiative. And if you look at this, you can say, well, these cells are not very similar. It's not very good at all. You have pretty significant quantitative differences in simulated drug responses between the adult myocytes over here, which is what we care about because these are the patients that the drugs are going to be given to, versus the in vitro system, which are these cardiac myocytes derived from stem cells. So this is a simulation of a selective blocker of the hERG channel, of the rapid delayed rectifier current. And the prediction is that this increases the action potential duration by 43% in the adult myocyte, but only by 22% in the stem cell-derived myocyte. And so if comparing drug effects in the simulations and the experiments on the stem cell-derived myocytes is an important part of CIPA, a pessimist would look at this and say, well, they're not very similar at all, right? Maybe they're qualitatively similar, but there's pretty substantial quantitative differences. So what we thought about when we were considering this problem is maybe we can use the models to help with this. Maybe we can use the models to correct for this. More precisely, drug effects can be quantitatively translated from one cell type to another through a model that combines the population-based mechanistic simulations and the machine learning. So using a strategy similar to what I've been talking about in this webinar. So what do we mean when we talk about population-based simulations to incorporate variability? I've kind of waved my hands about this and alluded to it a little bit, but this actually describes what I'm talking about. We impose heterogeneity in these populations by randomly varying the parameters that control the ionic channel abundances. So here's an example of a simulation of voltage and intracellular calcium from an adult myocyte model. We can vary the parameters and show that we get a slight prolongation of the action potential, a slight reduction in the intracellular calcium. Then what we can do is we can quantify all the metrics that I've already talked about. These are the things that in the experiment, if you had the cell in a dish, these are the numbers that you would extract from your time courses that you were measuring. And we can put those numbers into a vector here. So the results from cell 1 would be that some of these things are higher than they were in the control cell, and some of them are lower. So that's what we're expressing over here with this bar graph. Some are bigger, some are smaller. And then we can measure a second cell, and then we can quantify the metrics that come out of that cell like this. And then if we run the simulation over and over and over again, we can quantify the metrics from each cell that we simulate, and we can put all the results into a matrix that looks like this, where the heat map represents, if it's white, it's basically the control level, if it's red, it's below the control level, and if it's blue, it's above the control level. We do this over and over and over again. We do the physiological metrics quantification, and we do this for both the adult myocyte and the stem cell-derived myocyte models. So I'm going to talk about the results from the paper that we published earlier this year, but we've been doing this population-based simulation for a long time, for almost a decade now. And there's other references listed down at the bottom. So what we wanted to test in this study was whether we could build a regression model, use machine learning to build a model that would translate from one cell type to another cell type. So what we have on the left here is a matrix of the responses that we measure in the stem cell-derived myocytes. And what we have over here on the right is a matrix of the measurements that we would get from the corresponding population of adult myocytes. And what we need to relate this matrix to this matrix is another matrix in the middle, where if you take the stem cell-derived myocyte simulation matrix, multiply that by this matrix that we call B cross, well, you get something that is a good approximation of what you get in the adult myocyte. Because if you build this regression model and it works well, then what you can get is you can take an individual cell, measure the responses, experimentally in the stem cell-derived myocyte, multiply that vector of responses by B cross, and then you're going to be able to generate the predictions of how that same drug would work in an adult myocyte. So can this approach quantitatively translate drug responses across cell types and thereby overcome the experimental model limitations? And the limitations are what I showed a couple slides ago, namely that this action potential and this intracellular calcium look a lot different from this action potential and this intracellular calcium. So does the regression model predict drug effects? We have the stem cell-derived myocyte model here. Remember, we're going to simulate the measurements in the stem cell-derived myocyte model, multiply by our regression matrix B cross, and we're going to predict what happens in the adult myocyte. And these are the metrics that we're quantifying and that we're predicting. So if we take a drug that blocks the hERG channel, blocks IKR by 50%, this is an example of what that drug does in the stem cell-derived myocyte, prolongs the action potential, causes a very, very tiny increase in the intracellular calcium, but a prolongation of the intracellular calcium time course. And we plug that into our B cross cell type regression model, and we predict this is what's going to happen to the adult myocyte action potential. This is what's going to happen to the adult myocyte calcium. And because we're predicting these discrete features of the time courses, that's why our prediction in this case is discrete discrete points. That's why it's dots rather than a whole time course. But then what we can do is we can run the simulation and say, how did we do? How good was our prediction? And in this case, we run the simulation, and the solid line here is what you get from actually solving the differential equations. So what we can see is the prediction of the regression model, which is the discrete dots, is a very good approximation of what we get when we run the simulation. So the answer is yes, the regression model can predict drug effects with good accuracy quantitatively. We can do this a little bit more systematic. That's just one example. We can do this a little bit more systematically by looking at 30 drugs for which block of 6 ion channels was measured a couple years ago in this study by Crum et al. We simulate these drugs in a heterogeneous cell population where we're accounting for the fact that when we apply the drugs to the cells, not every cell is going to be exactly the same. So what we're looking at here is what the estimated drug response is and what the actual drug response is in an adult myocyte. And what we're plotting in green, or bluish green, is what you would get if you just made the assumption that whatever I measure in the stem cell-derived myocyte is the same as what I'm going to get in the human myocyte. So this is an example of amiodarone. Then we take the results we get from amiodarone, plug it into our regression model to correct for the limitations of the stem cell-derived myocyte, and we see that we end up closer to the line of identity, which is what we want. We want the estimated response to be the same as the actual response in the human cells. This is amiodarone. Rufinamide is another one where the estimate that comes just from the stem cell-derived myocytes is okay, but it's not great. If we run it through the machine learning multivariable regression model, we get a better response. And if we do this for all the 30 drugs that were measured in this study, we can see that these purple dots end up closer to the line of identity. We get a much better prediction than we get with the bluish green dots, which are a lot worse. So in other words, the cross-cell type modeling can identify and correct the mismatch that we have of these complex drug effects between the cell types. So to sum up this study, we think this is yet another example of how this how the mathematical modeling can be used in a more powerful way in the SIPA initiative, in the Comprehensive In Vitro Polyarrhythmia Assay. Whereas instead of just making measurements in stem cell-derived myocytes and comparing those measurements to what you get in the adult myocytes, or comparing those to the simulations, you should use a cross-cell type model to translate from one to the next in order to correct for the limitations of the experimental model. Okay, so in the time that's left, I'll try to wrap up in about 7 or 8 minutes so we have time for questions. I want to talk about some unpublished work in progress where what we're doing is trying to identify susceptible subpopulations. So this is work that's ongoing, and it's being led by Mira Varshnaya, who's a PhD student in the laboratory. The question we're trying to address here is closely related to the questions that we've been talking about, but we want to look at which individuals. So if you have a population of cardiomyocytes, and if this works, we can extend this strategy to actual people. What experiments would you conduct in order to predict which individuals are susceptible to arrhythmias? And as I already mentioned, the first thing that you look at is the action potential duration. This is why you do a thorough QT studies. If you want to look at which drugs are dangerous, you can look at how the drugs affect the QT interval. Well, if you want to look at which people are going to be susceptible, you can look at how the drugs affect their action potential duration. So this is the most common measurement that you would use to predict risk. And with the paradigm being that longer action potentials indicate more susceptibility, and shorter action potentials indicate less susceptibility. So D is for duration here, so we're talking about action potential duration, which is, again, analogous, that correlates with the QT interval. But it doesn't always tell you the whole story. You can have cells with the same action potential duration, but different levels of susceptibility. Here's an example from our simulations of a large population where we can extract 4 action potentials with the same APD. We can apply a perturbation, and what this perturbation is going to do is it's going to induce arrhythmic behavior in some of these cells, but not other cells. So what's happening here is this perturbation is prolonging the action potential, but what you can see is in these 2 cells, the purple one and the yellow one, it's inducing these so-called early after depolarizations. This is arrhythmic behavior, multiple after depolarizations, one after depolarization in the red cell. But it's only prolonging the action potential in the blue cell. So the blue cell is a low-risk cell, whereas the red cell is a medium-risk cell, and the purple one and the yellow one are the high-risk cells. So the goal of what we're trying to do now is to say, in addition to action potential duration, what are the other characteristics we can uncover that can distinguish these populations? How can we tell, just based on things you can see clinically or things you can see experimentally, before you add the perturbation, can you distinguish between the healthy and the arrhythmia-prone cells? So the research strategy is we build a large population of action potentials. We apply a trigger that might induce an arrhythmia in this population. And I'm going to show results from a couple different ones. But these are different ways that in these cellular simulation experiments, we can sometimes induce arrhythmias. You label each action potential based on whether it forms arrhythmic behavior or whether it doesn't. EAD here stands for early after depolarization. That's just a term we commonly use in the field. What that really means is arrhythmic behavior. You measure the metrics that we've been talking about. What are the things that as an electrophysiologist you would quantify? You use this combination of the labels as well as the metrics you extract. You plug those into different machine learning algorithms, and we're using a whole suite of them in this study. Then you determine which algorithm made the best prediction. So this is a more elaborate schematic here of the strategy that I outlined at the beginning for how we generate a heterogeneous population, analyze that heterogeneous population results with machine learning, and we're going to try to gain knowledge through that process and then through the feedback. So I just want to finish with a couple of examples of what we've gotten so far using this approach. For one thing so step one is we calibrate the population based on experimental data. We only want to take the action potentials that look like they're the physiological range. So we remove some of the ones that at baseline look kind of crazy. Then we apply a trigger on the population. The results I'm going to show are where we increase the L-type calcium current, and some of the cells form arrhythmic behavior, some cells do not. And we split the population into two groups, and we pick the size of our trigger so that we have a balanced data set where just about half of them gave us arrhythmias and the other half did not. And what we can see when we analyze this set of pseudo data from the simulation is that if we just look at the action potential, that can separate groups, but there's room for improvement. So if we just plot the resistant cells in blue and the susceptible cells in red, you can see that the susceptible cells tend to have a longer action potential duration. But there's a large group of cells here in the middle where the prediction could go either way. And that's what we want to use the machine learning to do better with that group in the middle. So what we see here is machine learning with two features: action potential duration measured at 50%, action potential duration measured at 90%, gives us decent classification. About 65% accuracy in an area under the curve of around 0.7. But then if we add more features of our action potentials and more features of our calcium transient, again, these are all the things that we would measure when we have the cell in a dish, we see that our accuracy increases. We get much better prediction in this case. We use support vector machine and logistic regression in this case. So better prediction when you give the machine learning algorithm more information. And people always want to know, we wanted to know what algorithms are going to work the best. We have a pipeline set up now where we take our set of data and we run it through all the different algorithms that are out there: random forest, k-nearest neighbors, logistic regression. We generally get very similar results no matter what we use. When we started this, because deep learning, artificial neural networks, convolutional neural nets are so hot right now, I had the hope that this one was going to end up being much better than the other ones. It turns out to be just as good as using logistic regression for the most part. There might be some examples down the road where the deep learning ends up being much better, but for the time being, it's about the same. So I just want to conclude by saying, what are the things that we're trying to address here? What are the questions we want to get at? I don't have answers for these. This is very much work in progress. I'd love to get some feedback. I'd love to get some suggestions. But this is to give you an example of the direction that we're going. Does machine learning performance remain the same no matter which trigger is applied? Does the performance change based on the parameters that are varied in the model? And then the most important thing in terms of feeding back for experimental prioritization is what other features about the baseline population can be calculated in order to improve the machine learning performance. So question 1, does it remain the same no matter which trigger is applied? The answer is no. I'll just quickly go through the results here. If we increase calcium or if we inject current, we get pretty good prediction, better prediction for the injection of current. If we block the hERG channel or we subject the cells to hypokalemia, which can be a proarrhythmic trigger, it's not nearly as good. We don't really know why. This is something we're actively addressing, but it turns out that the performance of your machine learning algorithm here depends on exactly what you're doing to the cells. This will help us understand, maybe it has to do with how the different mechanisms are represented in the models, and maybe this will give us a way to improve the models based on whether a particular perturbation is well predicted or poorly predicted. Second question, does machine learning performance change based on the parameters that are varied in the model? This is something else that we think is really cool, although we don't totally understand it now. These are 3 different categories of parameters that we vary in the model. Either we vary everything, which is what I showed first, which is the light blue line here. We only vary the maximal activity of these ion channels, proteins, these pumps and transporters. We only vary their kinetics, or we only vary how they depend on voltage. And what we see is for this purple line, when we only vary how they depend on voltage, we get much worse prediction than we do for the other cases. We don't know the answer why yet, but this also gives us something that this also gives us a way to feed back to the model to really dig in and say, well, what is it that's so special about generating the population this way compared to generating it the other way? What is it that makes this population more difficult to predict? And then the final result slide here is what are the features about the baseline population can be calculated to improve machine learning performance? We don't know what the answer to this is yet. The question that we're trying to address is, if you give more experimental protocols to say, I'm going to test my cells under different pacing rates, under different conditions, which one is going to be the secret that's going to improve the machine learning performance? And we're going to hopefully learn a lot about experimental prioritization that way. So this is, to wrap up, this gets back to our schematic, where we start with the simulations using these mechanistic quantitative systems pharmacology models. We're simulating a heterogeneous population. We're adding a trigger. We're distinguishing between the arrhythmic ones and the non-arrhythmic ones. We're performing machine learning to differentiate between the high-risk population and the low-risk population. And then what we want to do is we want to feed that back in order to return to the mechanistic models to figure out which experimental protocols are going to be the best ones, and whether there's something in the model that we didn't do so well initially, and improve our model through this process of feedback between the machine learning and the mechanistic model. So I'll finish by acknowledging people again. This is a picture of my lab from earlier this summer. In particular, Jingxi Gong, Mira Varshnaya, and one of the lab alumni, Megan Lancaster, acknowledge some collaborators here, as well as the sources of funding. So thank you for your attention, and I'm happy to take questions.
Thank you. Thank you, Eric, for a really interesting webinar. And just to remind everyone, you can enter your questions into the questions box on your GoToWebinar side panel. We do have a few questions, and we'll start off with the first. When you're in your slides, when you build a population of action potentials and introduce heterogeneity, is it possible to introduce the heterogeneity based on the therapeutic area or the target that you're looking at in terms of the drug?
Yeah, that's a good question. And the answer for the most part is we don't have nearly as much information at that level than we do at the level of, say, clinical measurements such as QT interval or contraction, or at the cellular level action potential and intracellular calcium. So yeah, one way you can introduce heterogeneity is if you knew what all of your proteins were in your cells, and you had made measurements of how those proteins, what the distribution of those proteins was from individual to individual. That would be a way from building the population from the ground up. But those are experiments that are hard to do. It's hard to, not that many people are interested in doing those kinds of descriptive measurements. So there's really not a lot of data out there. And so what we do is we do have large data sets on how, say, the action potential will vary from person to person or from cell to cell, how the intracellular calcium would vary. And we use those, we use those as our calibration procedure, where we make a large population where some of them fall outside the range, and we only restrict it to the ones that are in the physiological range. That is something that we're interested in. We're doing some work along that, but it's going to be a while before we know what it is for all the important components.
Thanks. On your project 2, could you talk a bit more about what your selection was for your regression model, and were there alternatives?
Yeah, that's a good question from the technical point of view. The regression model that we built was using partial least squares regression, and the reason we use partial least squares regression as opposed to other approaches was that we have a lot of outputs. We're going from the different things that we measure in the cells are going to be linearly correlated with one another rather than necessarily linearly independent. So regression is easy to do if all of your predictors are independent, but if they're correlated, or if you're not really sure how much they're going to be correlated, then the regression, straightforward regression can be tricky. I will say that yeah, some of this is in the supplement or buried in the paper. Jingxi went through a lot of work to figure out, well, how many components do you use in your partial least squares regression? This is a iterative technique similar to principal components analysis, where you can stop at a particular point depending on whether you're gaining more information or whether you're losing information and overfitting. And then the other thing that she did a lot of was to say which experimental protocols is a minimal set that's going to give us a good prediction. So we didn't look at that many other alternatives. We could have looked at lasso or ElasticNet, other things like that, which we didn't do so much. But we did spend a lot of time to address how many components or how many steps should we take in the partial least squares regression, and what should go into the initial matrix. That doesn't include everything that would be infeasible to do experimentally, but what would be something that is going to be predictive, but also something that could, in principle, be measured in realistic terms.
How do you perform the feature selection in your models, and have you tried engineering new features?
Yeah, that's one of the directions we want to go with especially with the last part of the project, where we have the population, where we divide the population into susceptible and non-susceptible. And yeah, that's a really good, that's a direction that we're really excited about, we're interested in. We're not really sure what the right answer is at this point. One of the things that we've, our machine learning expert has proposed are autoencoders, where we just feed the machine learning algorithm the entire time course, maybe as like an image, and we ask the machine learning algorithm to sort of pick out the features itself. There might be something subtle in the time course that you don't necessarily think to measure experimentally, but might be important in distinguishing the populations from one another. We've tried a couple things. We've really only scratched the surface. So far, it hasn't worked yet. And so what we're going with as our baseline is the things that I was trained as a classical electrophysiologist, the things that I was trained to measure. Basically, what the consensus is in the field for the features that you would get out of that. But I agree that this is where another area where the machine learning algorithms can be really powerful, and there's a lot more that we can do to automate that and make it less dependent on bias, less dependent on what the conventional wisdom is, and more data-driven.
So I'll be honest, we have a lot of questions. I'm going to try and summarize a number of them into groups. So there are a lot of questions about the ROC curves and whether they're based on test set or training set data, and whether they accurately predict the results, and whether they might vary depending on different aspects that you might use. So can you talk just briefly about the rock curves and some of the variations you might have looked at?
Sure. Let me go back. We did the rock curves with leave one out validation, which I understand is not great, but it is also, we only had 67 unique drugs, 86 drugs in total. I mean, maybe it's not actually very useful to go back and show them. This was done with leave one out validation. The other thing I would say that we have in the paper, in one of the tables or one of the supplementary tables, is we looked at a few other measures of prediction strength besides just the area under the rock curve. One of them I think is called the Matthews correlation coefficient, and the other ones I can't even remember what they're called. But yeah, we did look into a couple different ways of assessing the classifier performance besides just the area under the rock curve, which I know gets a lot of criticism in the field, and they all correlated with one another. Although I also will acknowledge that because we didn't have a very large data set, we couldn't do things quite as rigorously as you might want. It was hard to divide it into half, for instance. So there are some limitations there.
And that leads into one of the other questions, is whether or not your different machine learning techniques might have been improved by either more data or by analyzing the entire curve rather than discrete points.
I would say that's an active area of research for this study, the drug classification study, where we again only had 86 drugs, or 86 or 67 depending on how you counted. But the reason I keep going back and forth is for a drug like dofetilide, you'd have one set of measurements of the IC50 values, and you'd have another one. So we used both of those different sets of measurements. Because we had a limited data set in this study, we didn't want to push it even farther by looking at questions like looking at the whole curve, or really looking at too many different algorithms, et cetera. But for the newer study, for the unpublished work, where we're looking at susceptibility of a population, and we can always add more individuals to our population, that's where we think that there's promise in looking at, say, the entire time course, rather than just the discrete features. So that's work that's being done right now, at this moment, and hopefully we'll have some answers soon. But I agree with the general tenor of the questions, where there's more that we can do, and I think that doing more is gonna give us additional insight.
And do your risk markers identified by the machine learning depend on the cell model?
That's a good question. We did, this is something I kind of glossed over in the drug classification study, where we looked at multiple models, because we tried to take an agnostic approach, saying that there's different groups have developed these cell models, and they're all very serious, very rigorous scientists. So we're not going to go in thinking that one is better than the other one. Let's just test all of them and see how it comes out. I would say without having the data right in front of me, I think the answer is yes. I think that one of the three models we looked at doesn't do as good of a job as the other two at simulating intracellular calcium. So that one would probably tell us that the intracellular calcium is not important, and that would represent a model flaw, more likely to be a model flaw rather than something that's actually reflective of the physiology. But that's something we've looked at a little bit, but not maybe as systematically as we could have, to see how the different which model that we're looking at tells us which features are important. Sorry I can't be more specific than that. I think that, I think the answer in general, though, is going to be yes. It will depend on the details of the model.
Thank you. Just to let you know, we do have quite a few questions left. I think that more than could possibly be addressed in this time slot. We will forward, for the audience, we will forward your questions through to the presenter to address. With that, I want to thank Eric for a really interesting webinar. We have gotten quite a few thank yous and comments saying what a great presentation this is. So with that, thank you, Eric, for a fabulous webinar, and thank you to our audience. And I hope to see everybody again at a future webinar.
Okay, and I scrolled back to the title slide so that everyone can see my email. I'll answer the questions that get forwarded to me, but I do encourage people to contact me. I'd be happy to talk more about a lot of these issues because I know that people are thinking about similar things, whether they're looking at arrhythmias or whether they're looking at other questions. Probably more people on other questions rather than arrhythmias. I know that there's a lot of interest in the field on these sorts of questions, and by talking to each other, we're going to come up with better solutions. So I encourage that. And I thank everybody for paying attention.
Thank you, Eric, and have a great day, everyone.
Okay. Thank you.
Bye.